My name is Griffin O'Neal-Freeman. I've been working in the Corwin lab this summer, and today I'm going to present experimentally studying extreme diffusion by centrifusion colloids. So what am I going to talk about? Firstly, I'm going to talk about diffusion, what it is, and two models physicists have come up with for it. Then I'm going to talk about extreme diffusion, what the word extreme means and why it's important. And lastly, I'm going to talk about experimentally centrifuging colloids, which is the work I've been doing this summer. So what is diffusion? Diffusion is things spreading out. That could be oxygen molecules in the air. It could be microbiological zebrafish gut bacteria, or it could be COVID cases in Italy from February 25th to March 25th of 2020. So this phenomenon of diffusion is really widespread. It's happening all the time, all over the world. And because it's so common, it's important that we come up with models for it so we can predict the behavior of diffusing systems and use those predictions to solve problems, like how fast will COVID spread. So I'm going to talk about two models today, and both of these models hinge on a concept called a random walk. So what is a random walk? Particles move in random walks. So this figure is to help me explain what a random walk is. In this figure, you can see a simulated dust particle suspended in water. The gray circles are water molecules, and the colors are arrows showing the flow field of the water. So Haley talked a little bit about Brownian motion. And as you can see, the black line shows the path of the dust particle with time, and it moves sort of randomly and erratically, and that is due to that Brownian motion. So why is this important for a model of diffusion? Well, the person who created the first model I'm going to talk about was Einstein. And he looked at this and thought, okay, I can model a diffusing system by treating each diffusing object independently and saying that at any given position, it has an equal chance to go in any direction that it can go in. So what does that actually mean? Let's look at a one-dimensional example. This is a one-dimensional diffusing system with discrete positions and discrete time steps. The positions are the blocks and the time steps are also the blocks. Here you can see that all the particles start in the middle. And then because this is a one-dimensional system, they can either go left or right. They have a 50% chance to go in each direction based on Einstein's model. So what happens is half of them go left, half of them go right, and then that happens again and again until eventually you get a Gaussian probability distribution. And this model is called simple symmetric random walks. You can see the symmetry. So that's the first model. The second model I'm going to talk about hinges on the idea that particles that start next to each other move similarly. So here we have the same situation, dust particles suspended in water, except now there are blue and red dust particles as well. And so the idea here is that if you were to have two particles that started near each other, and you can imagine that they would be similarly affected by the flow field in that environment if they start close to each other. And so this idea actually works. As you can see, the particles move in similar paths. The blue line is pretty much identical to the red line. And so why is that idea important? Why is the idea important that diffusing objects that start near each other will move in similar ways? This is the other model I'm going to talk about. This model was made by Jilami Baraquand and Aileen Corwin. And in this model, you can see that the blocks are colored, and the colored blocks represent bias from the flow field. So red blocks are biased to go to the right, and blue blocks are biased to go to the left. And so it looks a little bit different. The diffusion in this model is not symmetric. You can see there are some more particles a little bit on the right side. And so this model is called random walk in a random environment. The colored blocks being that random environment. And so this random environment not only changes with position, it changes with time as well. So this is the second model. And in this figure, you can see the models side by side. And again, you can see they look a little bit different. The Einstein model is very symmetric, whereas the Barraquan-Corwin model is not symmetric and looks a little funky. So why are these models important? Well, it turns out that these models have similar predictions for where the bulk of diffusing objects in a diffusing system will end up. So what do I mean by that? In this graph, you can see probability density on the y-axis and position on the x-axis. And this is the graph for a diffusing system in the simple symmetric random walk model. The y-axis is on a log scale, which is why it doesn't look like a Gaussian distribution. And the line is smooth. Now, this is the same graph, but for the random walk in a random environment model. And as you can see, the line is spiky, and that's due to those biases at different positions. And if you look at these, you can see that they look, about the same. The random walk in a random environment one is a little bit bumpy, but it doesn't really matter for most of them. But if you look at the tails, that matters. The tails are very different. Depending on the different spikes at the tails, the diffusing objects could be predicted to be in the same place or very different places. And the Corwin lab has done some theoretical and numerical modeling, and it turns out, based on our predictions, these two models have different predictions for the behavior of the outliers of a diffusing system, the behavior of the extremes of a diffusing system. So that's the main takeaway here. Outlier behavior is predicted to be different between these two models. So it's predicted to be different, but is it actually different? And that's what I've been working on this summer. So the next step is to do an experiment or experiments to see which model is correct. So the basic idea for doing this experiment is using a one-dimensional system to simplify things as much as possible. And how this works is you start with all your diffusing objects on one side of the system, and then you allow them to diffuse across the system, and you measure how long it takes for the first object to cross a certain distance. You measure that time. And then you do that again and again, and you build up first passage times. And you can do statistics and analyze those first passage times, and then compare the experimental results with your predicted theoretical and numerical results, and see which model is right. So what does this actually look like in experimental form? Well, okay. Well, to create a one-dimensional system, we use very thin capillaries, about 0.5 millimeter by 0.05 millimeters. Capillaries are just tubes. And so that's to get as close as we can to a one-dimensional system. And then we need a diffusing object. We need something that moves around. And so we use colloids, which are 1 micron silica particles. And so you get the colloids in the capillaries, but then you have to get all the colloids on one side of the capillary. So how do you do that? Well, our idea is to centrifuge the capillaries. So here is sort of a basic image of the setup. You have your centrifuge stage. And so you put your capillaries on the centrifuge stage. The centrifuge spins. All the colloids go to the outside of the capillary. Then the centrifuge stops spinning, and the colloids diffuse back inside the capillary. You have a microscope camera set up at a certain position. There you can see imaging objective, that's the microscope camera, and excitation light. There's light that's lighting up the capillary from below, so you can see what's going on. And you're taking pictures at that certain position. And when the first colloid comes into frame, you measure the first passage time, and you do that again and again, and that's how you build up data, right? So this is the basic experimental setup. This is what it actually looks like. I apologize for the crappy image. So this is the centrifuge plane. You can fit 16 capillaries at any time on the centrifuge plane. And there are little white dots on it. That is wax holding down the capillaries so they don't fly off during centrifugation. This is the motor down here that spins the centrifuge. This is a feedback control system for the motor that controls the speed that the motor spins at. And this Arduino controls the motor speed and the feedback control, and that's connected to a computer. The microscope objective is shown here. And so what happens is the light illuminates the capillary, and then that light goes up through the microscope objective and into the camera, and the camera takes a picture. The light is powered by this box here. And once again, the camera is connected to the computer. And via the computer, we use MATLAB on the computer to control this whole experiment. So we have a big code file, and we can run the whole thing from there. So this is a picture of a capillary with some colloids in it. Thought it would be nice for you to see it nice and big. All the dots are colloids. There are lots of dots on the bottom, lots of colloids on the bottom. Those are free-moving colloids. The colloids that are higher are stuck to the glass. So that's the first problem I want to talk about. This was unfortunately not a smooth sailing process, but I don't think experimental physics is. And so the first issue is sticky colloids. What is happening is sometimes colloids get stuck to the glass. Well, pretty much every time they get stuck to the glass, and they come off early because they didn't go to the end of the capillary, and then give you bad data. Another problem is blurry photos. I have 16 capillaries on the centrifuge, and some of them are in focus and some of them aren't. And if they aren't, you can't see the colloids. And so we would like them all to be in focus. Another problem is bubbles. Sometimes you get air bubbles, and what the bubbles will do is they'll push the colloids down the capillary in what is called, forget the word, but not diffusion. That is so that is also bad data. And then bulk flow. And then another issue is the distance of the camera from the end of the capillary. If that distance is too big, we might get a very large diffusion time, a very large first passage time, so large that it's pretty much useless to try to measure it. So we've had some problems this summer, but we've also come up with some solutions. So one solution is bovine serum albumin. This is a protein that comes from cow blood. And what this helps with is if you put it in the capillaries, flush it out, then put the colloids in, the bovine serum sticks to the sides of the capillary, coating the walls, and helps to prevent colloids from sticking to the side. To deal with the focus issues, we haven't done this yet, but we could make an automatic focus adjuster and connect it to the microscope focus adjuster. This would allow us to adjust to the best focus for each position on the centrifuge and take good pictures. To deal with bubbles, I found that after I made the capillary, I just had to really carefully examine it. It's kind of hard to see at first, but with practice, you get good at it. And you just snap off any parts that have bubbles. They're usually at the end. And then to deal with the distance of the camera to the end of the capillary, we can use a translation stage. So what does that mean? Well, it turns out we had this graph showing colloid first passage time versus path length, which is how far the colloid has to travel. There's diffusion time in days on the y-axis and distance from the origin on the x-axis. And we measured this distance recently, and it turned out that it was a centimeter. And so that means a diffusion time of about 41 days, which is not great. So a solution to this is buying a translation stage and putting it under the microscope. That way we can move the microscope and change that path length to hopefully make it something like 5 days or 3 days. Yeah. So overall, in this presentation, I talked about diffusion, and I talked about two models, simple symmetric random walks and random walk in a random environment. I then talked about extreme diffusion, And the main takeaway there is that outlier behavior is predicted to be different between these two models. And lastly, I talked